Okay, thank you very much, Dennis. I'll try to keep the time. Firstly, thank you to the organisers. It's very nice to be back here again, but as Dennis said, we're now in the sort of the world of the future, so I'm going to make no references whatsoever to what I was doing 20 years ago, which is fine by me. So my title here is New Methods and Future Challenges. When you're asked to give talks like this, of course, it's always very difficult to know how to pitch it, partly because you can't really predict the future, but also because there's a sense in which you end up really being sort of rather selfish, and let me sort of illustrate what I mean by that. So many of you may have seen this little illustration before. So this is how professors spend their time, this is from some survey sort of over 10 years ago. So teaching, research, a sort of administration like this, and of course the key thing about this is in fact this says, don't tell me what to do. So I'm going to carry on in this vein by essentially assuming that the future kind of revolves around me and my interests, but having said all that, I'm pleased to say that on the basis of what we've seen so far, there's going to be some overlap. I'd also be very happy for people to chip in with comments as we go along, and as Dennis has emphasised, this is we should really be having a bit more discussion now. So I'm really just going to sort of flag up a couple of areas which I think are sort of very important in the future and which have already begun being important in the sense that people are already actively working on them or starting to work on them or starting to think about them in some detail. So the first of these is what I've called high-resolution genetic or maybe even genomic data, and the second one is model assessment. And of course we've already in the meeting so far heard a little bit about both of these, but I want to say perhaps a bit more detail about some of them. Okay, so this is the one of the pictures you will have seen before. So if you go onto the website for this meeting, this is a picture that Chris had this morning. So this is taken from this paper in New England Journal, and something this is to do with an analysis of an outbreak where there was sort of genetic data involved. So this is in a sense showing us something about the way ahead in this area. So let me just sort of say a bit about what I mean by this. Now, I should, of course, apologise by saying I'm not really an expert in this area, and I don't really know all that much about biology and genetics and genomics and so on and so forth. So if you know more than me, that's fine. Please pipe up and tell me things that are wrong. But this is the kind of thing I have in mind and how it relates to the sort of things I'm interested in. So first of all, when we talk about these kind of data, what do we mean? So usually I'm talking about things which are individual level data on the pathogen. All right, so the very obvious thing to say is that we talk about individuals in a population, so typically humans, it could be animals of course, but rather than sort of keeping track just of them, the usual epidemiological quantities of when they're infected and cases and so on, we actually have data on the pathogen. Data like this could come at sort of single time points or multiple time points. So for example, single might be if there's a situation where you identify a case and then there's some sort of sampling which takes place, and then we have just at that point in time some information about their sort of genetic information, if you like, or possibly multiple time points. So in some contexts there'll be sort of longitudinal data of the same individual through time. And of course, as soon as we sort of see this kind of thing, we might be thinking that there are issues to do with sampling, and what is it that we're sampling, how are we sampling it, what are the statistical processes going on, etc., etc. So immediately there's something which will cause us some cause for thought. These things are very high dimensional. So the sort of example we have in mind, or I have in mind, is like whole genome sequences, and so typically you're looking at a very, very large items of data, sort of very high dimensional information that's sort of coming in here, and we'd like to know something what we do about that, and that causes some problems, of course. Now this is something that as people who work in this area will know all about. I don't think it hasn't specifically been maybe mentioned too much so far, but there's two sort of very different areas in this field. So one is what you might call high frequency sampling. So this is more to do with an outbreak of disease where we collect this sort of high resolution data, and most of the cases that we see, or ideally all of them, have that information associated with them. And of course that's a sort of useful and could perhaps tell us something about how the disease is spreading in particular. And then in contrast, there's what's called low frequency sampling, which is more the sort of thing where you're just going out and maybe doing a survey across a population. So you're certainly not sampling everybody, and you're certainly not sampling all of the cases through time of a disease, for example. So these are very, very sort of different settings, and of course they have very different challenges and so on. And as you know, this sort of thing is becoming far more common, basically due to sort of cost reduction. So here's a picture of a sort of desktop sequencer, and the sort of things that you might get out of it. All right, now the next question is, well, so what? What use are these data? So what do they really tell you? And as has already been picked up in the meeting, there's sort of two things going on. So there's an epidemiological side. There's also a sort of, if you like, pathogen evolution side of things. So what can they tell us? From an epi side, the natural thing is to say this, that if just I observe more information about the pathogen as it goes from, say, individual to individual, surely that must tell me more about who is responsible for infecting who. Okay, so that's the absolute sort of obvious thing to say about this. And you know, that seems to make a lot of sense. Now, you might also think that this automatically means that you get better estimates of your epidemiological quantities, like R0 or transmission rates and so on. Okay, now you'll notice I've put this question mark here, which indicates that this isn't necessarily always true. All right, so we've even looked in our group at some very, very simple modeling examples which show that it's perfectly possible to have, you know, situations where if you observe these extra data, they don't necessarily reduce or sort of improve, I should say, the accuracy of the estimates that you get for things like infection rates, which is slightly counterintuitive, but if you sort of think through what's going on in terms of you have more complex models, there's more parameters, so maybe it's not as bad as all that. Of course, the other side is that you may also want to understand the evolution of the pathogen. So this is sort of the opposite side of the coin, saying, well, I'm not so bothered about, you know, the disease spread, but I'd like to know what's happening sort of on the genetic side or the genomic side. So this is a sort of data set that if you look in the high frequency sampling case you might get. So this is a data set on MRSA, or if you're from North America, MRSA. And this is from a hospital in ICU in Thailand. And what's going on here is, so we've got time on the horizontal axis. This is the number of individuals on the vertical axis. And the sort of the red area is the population of individuals who were colonized, meaning that they had a sort of positive swab test for MRSA. This one is the population in the ICU itself. And what are these little lines and blobs? So each line corresponds to an individual, and each of these blobs corresponds to basically a sort of an isolate which was then sequenced. So you can see some of the individuals have like maybe two or three occasions where they get information taken. This one in the middle has a huge number. You'll also notice that they got different colors. What's that about? Well, that was essentially, it's one attempt at trying to sort of cluster things together and say what's similar and what isn't and so on. So, and even a picture like this, you can tell straight away that, you know, there's some instances where somebody's sort of genomic information doesn't really change very much, or the pathogen doesn't change much. Others, it seems to change quite a lot, and sometimes not at all. So there's all sorts of different things going on. So the kind of immediate challenge is to say, well, how would you deal with these kind of data in a meaningful way? Now, what are of course, the blobs in more detail will be something like this. So these sort of strings of letters, for those of us who don't work in biology. So these are like nucleotides. And of course, for something like MRSA, there's sort of about 2 million of these things. So it's like some huge number that we're trying to keep track of. So it's a very high dimensional problem. All right, now this is not slightly inaccurate slide, because I think this should probably say at least 2 kinds, but because I mentioned this was sort of, this is an O'Neill-centric talk, so I only really know about 2 myself, but there's probably more. So there's 2 ways you might try and analyze these kind of data. So, well, and again, these are just sort of some names of people who work in this area, and it's not supposed to be exhaustive and so on and so forth. So one thing you can do is to sort of somehow separate out the genetic and the epidemic components, so sort of treat them separately. The other thing you might try and do is sort of do it all at once. Okay, so somehow have a model which includes both of these things at the same time. So let's just very briefly think about these 2 different things. So the first one, the sort of thing that you might do, this is so some people do things like this, is that, and this is more what happens in a sort of low frequency sampling. Okay, so the situation where you don't observe the whole population, you know, all the time. So what you might do is, just given your information, you might sort of try and come up with some estimate of the phylogenetic tree. And then once you know what that is, or you think you know what it is, you then try and fit some kind of epidemic model. So some of the sort of coalescent theory and so on takes this kind of route. Another way you might do it is to sort of say, well, we already know how to solve problems involving, for example, strain type models, or we sort of try and cluster people together genetically similar groups. So what we might do is do that to start with, so just look at what there is and give everybody a label, and then once you've done that, just fit some kind of multi-type epidemic model. And the point here is that the number of types won't be all that large, as opposed to if you think about the real sequences, there might be sort of a very large number of types. So that's the kind of thing that people would do in that setting. Okay, so what's good and bad about this? So this is sort of discussion points, if you like. Obviously, it's kind of a simple approach, and it avoids having to do complex modeling. And what do I mean by that is that it really ignores any relationship between transmission and genetic information. So now, here, of course, here's a sort of a grey area, or certainly for me, because I don't know much about this, but the whole idea of how somebody's sequence or exactly what's going on for them affects the transmission. Okay, so there's that whole area of what's the interaction between those things? Are they how independent or otherwise are they? But this sort of approach sort of, to some extent, ignores that. So what's the alternative? So one alternative is then to sort of try to do things simultaneously. So now you might model. Now the problem here is that you now sort of model genetic evolution explicitly in the model and define a model to feature both of the genetic and epidemic parts. So just to be slightly more clear what I mean about this, in contrast to the first case, what I mean here is that it's a model, meaning that I could simulate it, say, on a computer. So I could, and as the model goes along, it tells me who's infected and who isn't and all this sort of thing. But it also keeps track of the sort of sequence information or whatever level of detail we've got. All right. So in other words, it's sort of saying, can I write down a sort of a model which evolves forward through time and simultaneously keeps track of all these different things? So the good news about this is that it seems like an integrated approach. Now, one of the sort of, I guess, points you might want to think about in terms of discussion is this. Is this kind of modeling too detailed? So what do I mean by that? Well, when we, sort of in classical mathematical epidemiology, we've over the years come to the conclusion that you don't really have to keep track of too much. So if you look at an individual, you know if they're susceptible, maybe they're latent or exposed, infectious. Maybe for different diseases you might have different prodromal period or something. So you can put in a few things, but at the end of the day, there's not that many types that if you're an individual going through time, you can't be that many sort of different sorts of individual at a particular time. Now here, what's happening is we're suddenly going to a situation where we're trying to keep track of potentially sort of millions of these nucleotides, and we're sort of saying, well, how do they evolve and what's going on? And that suddenly we've gone to a sort of very broad and easy to understand sort of we're happy with it level of modeling down to something very, very detailed, and also something very detailed which we don't really understand very well. So I'm slightly worried about that. And so, for example, there's lots of these models which, well, not lots, there aren't many, but some of the models which do this kind of thing are so-called microevolution models. So they will look at the nucleotides and say, okay, they're all evolving according to Poisson processes, and there's a Poisson number of mutations and all this sort of thing. And it's sort of, well, is that really true? Is that reality? Should you work at that level? What about recombination and other things and so on? So, so there's a whole interesting question about, is it too detailed? Now, one of the things we've done in our group is to instead have a slightly more abstract version of doing this, which I'm not going to talk about in detail, but I'll just describe for a moment, which is this, that when you do this kind of modeling, what you're really interested in is not necessarily what is everybody's sequence, but what you're interested in is how different are the sequences. So there's this sort of, and most of the way that the transmission models are built up will look at things like, let's count the number of what's called SNP, single nucleotide polymorphism, so the difference between two of these genome sequences that you've got. And if that number's large, there's more difference, and and it's a very, very crude sort of way of doing it, but that's what happens. But what you could do is, in fact, just say, well, all I care about is actually the differences between individuals. So actually try to model those differences and try to come up with a way of moving through time. And when new individuals come into the population, you're interested in the difference between them, or new infectives, and other individuals. Okay. So we tried to work in that way. The other thing I've mentioned here is what's initial conditions typical sequence. Again, if you have a sort of classical epidemic model like an SIR model, SIRS or something like that, and you allow people to come into the population, usually when they come in, we might say, okay, they're susceptible, or maybe if your model's very advanced, they could be susceptible or infective, or whatever it is, so there might be a bit of a choice. Now, if you're trying to model sequences, you've got a bit of a problem. You say, okay, they arrive, what's their sequence? how do you suddenly sort of come up with a way of proposing, if you like, or just describing what that is? What do you choose from? It's suddenly you've got an enormous space of possibilities. So there's a few sort of interesting questions around that, and I think this is certainly an area that we need to think a lot more about. Okay, just to finish, in another 5 or 10-ish minutes, yes, Dennis isn't cutting me off yet, okay. I just want to say a little bit about model assessment. So this is something that Simon raised yesterday, and I think is very, very important. So if you sort of look back through the history of analysing data in using infectious disease models, and particularly mechanistic models, what you'll find, of course, is that sort of back in the 70s and 80s, you're using maximum likelihood and trying to use a computer to sort of numerically optimize likelihood surfaces and so on. Then MCMC arrived and the sort of whole Bayesian thing, and all of a sudden we could do very complicated things and fit very complicated models, and that was great. And of course the advancements continued, SMC and other areas as well. But unfortunately, things haven't really kept up on the model assessment place. So it's very common now, and I mean I haven't deliberately not done this exercise for fear of insulting anyone in this room, but if you sort of read through lots of papers which fit models to data, it's quite disturbing how few of them make any attempt to see whether the model's any good insofar as does it fit, are there, is there enough evidence that it's sort of tracking how the data look and so on. I mean, there's some things in some papers, but I think that it's an area that needs a lot more attention. Okay, so what do we mean? So there's sort of two things. One is, if you like, model fit, and the other way you might think of as model choice, in other words, a comparison, maybe different competing models that you're interested in. Okay, why should you bother to do it at all? So I say it doesn't seem to bother many of us, but okay, well, I guess the main, okay, why not do it? Because it's easier to get your paper out quickly, but why do it? So poor fit sheds doubt on conclusions from modeling. I mean, this is the absolute sort of scientific baseline of what's going on here. You fit your model and there's the conclusions, and people say, yes, we've shown that, R0 is 58 or something like that, or whatever it is. And, but if the model doesn't fit, well, that doesn't seem very good. And of course, another reason to it is that model choice can be a tool for directly assessing questions of interest. So informally or formally, you might think of a kind of hypothesis testing where you say either transmission happens like this or it happens like that. I've got two models. I look at how likely both of them are relative to each other, or some measure of that sort of thing. Okay, those of you who are not statisticians, let me just sort of bore you for a moment with this. So in the world of linear regression, so this means you've got some data, like some dots. What we try and do is sort of draw a kind of best fit line through them. And so this is what's called a linear model. So second year statistics or mathematics. So you've got a couple of parameters, and you've got some sort of error term which you assume has a sort of Gaussian distribution. And what happens is you basically try and minimize the distance of the sort of the mean of this model from the observed data. So if you like, doing maximum likelihood estimation is equivalent to doing sort of like least squares fitting, essentially. So you kind of minimize these residuals, and that's the sort of thing you do. Now, of course, one of the problems with this area is that it's very hard to just sort of take those ideas and just put them straight across. So for outbreak data, one question is, what are the right residuals? So one thing here is that, of course, you have variation in time as well as in space as you move along. So, events don't always happen on a Tuesday or whatever it is, so there's different things going on. Should you use observed or unobserved data to compare? So this paper, I think, or some of this work was mentioned by Simon, so by George Streftaris and Gavin Gibson, where they look at sort of residuals of a certain kind, but based on things you don't observe in the epidemic model. If you want to look at what, following the ideas of regressions, you want to look at the mean, it may be that you kind of only get this by simulation, and for that matter, is even the mean the right quantity to consider. So the sort of thing you might want to do is things like this. So I thought this bright green color would wake up anybody who was just still a bit sleepy after lunch. So this is just to sort of show you what are these. So this is an SIR model, and there are three sorts of infectious period, either an exponential distribution, a gamma distribution, or a constant. And what's happening here is you've got some data which come from this model, which I think is the blue line, if I remember rightly. And what happens here is you try to fit you fit all these three different models, and you look at the means, and then the greens are sort of like a bit like Bayesian predictive distributions around that, conditioned on a particular final size. And the idea is that you might look to see, well, can I tell, if the data are from this model, can I tell which model they came from? So, for example, the constant, there's some discrepancy here. We're not picking up things at the bottom. So maybe there's some sort of mileage in this kind of model assessment in a sort of, trying to take some of the ideas of regression a little bit further. There's also lots of sort of simulation-based ways to look at model fit. So what quite a lot of people do is to just say, okay, I estimate my parameters, I then simulate the model lots of times, I plot that on top of my data and say, does it look similar or not? Very common way of doing it. But at the same time, you might have to decide what is it you're comparing, so the whole choice of summary statistics. And of course, all of this is quite closely related to sort of ABC methods. So TJ and Peter in the audience, and I might pounce on them to say something about this in the discussion, but there's another sort of interesting suite of ideas that one can use there. Okay, what else can you do? There's also things, here's sort of 4 other ways you might think of doing model choice. So hypermodels, Bayesian non-parametrics, Bayesian methods like reversible jump and mixtures. So let me just whiz through them. So what do I mean by hypermodels? So this is how do you choose between competing models. So let's suppose you want to say, I think the infection rate in my SIR model is beta S, or maybe it's beta SI, or maybe if I'm feeling ambitious, it's beta S times the square root of I or something, and I'd like to choose which of those 3 it is. So a natural way to do that is to build a model which includes all of them as subcases. All right, so that's the sort of, to my mind, it's a fairly natural way of doing things. So you just have a parameter and you try and estimate what it is. So that's one way you can go about model choice. It's sort of a kind of an easy way in the sense you just have a more complicated model. That's something that's worth exploring. Bayesian non-parametrics, something we've been doing a bit in our group at Nottingham, is to sort of say, well, if you have an SIR model or others as well, and this infection rate, you perhaps want it to vary through time, then there's a sort of machinery around there. So for example, using Gaussian processes, where you can try and do this. So you try and basically, you put a prior distribution on what you think the function looks like, and then you try and estimate it from the data. And it turns out that that's actually quite a nice thing to do, partly because it's possible to do it using MCMC, and well, actually it's possible to using fairly complicated MCMC, but fortunately this chap here is very good at programming. But also because it starts to give you some illustration as to whether your model fits or not. So it's just a sort of random picture of some simulated data, which is some dots and the fitting that we get in this case, which is quite good. Reversible jump is another sort of thing. I mean, this doesn't get a good press, and it's not very easy to do it. I mean, those of you who've ever tried to do this sort of thing, it's not easy to do. You can use it to sort of work out Bayes factors essentially. Okay, and the final thing I just mentioned, this is something that Theo Kyprios and I have been working on recently, is that it turns out there's another way of doing model choice, at least to get Bayes factors, which is to sort of basically take 2 models and just make a mixture model up like this. You just sort of say it's the chance of this model is alpha times this one plus 1 minus alpha times that one. And intuitively, if you fitted this mixture model and you estimated alpha, you think that must tell you something about how likely these 2 models are, right? If alpha's 0, surely then it must be this one here. If it's alpha's 1, it must be this one. And it turns out that if you can estimate the mean of alpha, you can actually work out the Bayes factor. And this kind of gives you a way of doing it without doing reversible jump. So, but this seems quite promising. So we're just working a bit on this at the moment, and we should have a paper out fairly soon. And I think we're in time for discussion. So thank you very much.
5 minutes discussion. Left leg. There's a bit of a middle ground in between separating the genetic information and the epidemiological information, because there are 30 years or so of trying to model how pathogens evolve using quite sophisticated codon-based models and so on. What you can do, if you do separate those 2 out, is you can always go back and think of those genetic models as a proposal for some more realistic models. So you could essentially go back and re-weight those models separately. So you could tie those 2 together a bit more.
Mm-hmm. Mm-hmm. Yes.
Jim.
So in terms of model assessment, it seems to me one of the crucial things is not is one model better than the other, but you have to specify what the inference that you're trying to make is. Mm-hmm. And you want to make sure your model inference is robust to realistic relaxation of the simplifying assumptions in your model. So that seems to be a more solid basis So it's focusing on the robustness of the inference, not the robustness of the model or the exact fit of the model. Because you may have, if you're either you're focusing on a hypothesis, like you said, between different modes of transmission, but more in the public health sense, I'm going to do this intervention, or I'm going to do that intervention, or I'm going to, wait between them. Yeah, at that, if you can get down to that level, first of all, we're going to relate to public health people better, because we're going to answer the questions that they want to focus. But also then, we'll be able to evaluate whether our model would in fact be robust or not if I realistically relax on the subject.
Yeah, no, I completely agree with that. I think that although, as I said, I sort of have some concern that we have a tendency to just estimate models' parameters because we can, and then just say, well, it doesn't care if it doesn't fit. But I think you're right in that what ultimately should drive it is what we're trying to achieve. So if there are particular questions that you want to answer or things you want to estimate, or intervention policies of vaccination or whatever that you're going to then say as a result of my estimation, this is what you should do, then I think you're right that this is exactly the sort of thing you want to be able to sort of say how robust is that, and and get some assessment of what's happening with that outcome. Okay. Thomas?
Isn't that kind of an issue that if you really are in a Bayesian framework, you ought to be doing your kind of hyper model, but just over, so you ought to be doing reversible jump, or if you could have a better algorithm, but you ought to be somehow averaging over some enormously big model space, and then produce these kind of green lines across that whole model space weighted by your full. And the issue is no one can do that technically, so all the rest of the model selection is a bit of a kind of trying to do something a bit unprincipled from a Bayesian point of view.
Well, it depends how Bayesian you want to be. And I think also there can be a danger of being a bit too honest. So, and I think this is a problem that has come along in Bayesian statistics before. So what tends to happen is that people fit models and say, oh, we get these MLEs or something like that, and everything looks very good, and the predictions look good, and the public health people say, yes, we like the look of this, it's all very robust. And then the Bayesians come along and say, no, you're not accounting for uncertainty properly, let's put some more of that in. And then before you know it, your predictions start spreading out and out and out, and the public health people say, well, these Bayesian methods are hopeless. When we did it with MLEs, we got the right answer. So I think one has to be slightly pragmatic. I mean, yeah, you're right. If there is a sense in which perhaps you should expand what you're looking at and sort of go through the machinery and so on. But I think one has to be pragmatic as well. Yeah.
If I could interject myself, isn't there slightly this idea that you just want to, doesn't really matter which model as long as the conclusion is reasonably reliable?
Yeah, well, that's right. I mean,
that's not the focus on the present problem. I think it still remains a useful question to ask. I mean, suppose two models fitted equally well in this case and give you the same estimate of what to do. It's still an interesting question as to which of those models really was matching the reality better, because there are going to be other sets of data and other diseases coming along, and you want to know which one to go with next time. So,
Aaron. On the same subject, I wonder, and coming back to your question, I wonder whether the effort that's required to really better assess model fit and compare models on the basis of their ability to explain existing data is better spent looking at might be better spent in many cases, looking at other sort of independent ways of assessing the model fit. So forecasting, for example, is something that people are recently, in relatively recent times, starting to actually try to do on the basis of even fairly simple models. And this gives you a way which is sort of independent of model fit of actually trying to assess whether the model's really good or not. There are other approaches as well, but I think perhaps rather than devise an extremely complicated algorithm, it might be good to spend that effort to explain.
Maybe time for one or two more questions, but Otto, we'd like to be getting set up. Paolo, we just had a last question or two.
A quick one on the same theme. Apparently model choice is an interesting deal. You mentioned already 2-3 methods, and then there are some more proposals. Does anybody know if they kind of give the same answer? If you try to interpolate the alpha, or you instead just compare the two models and succeed in getting a base factor, or if you do a third thing, will they usually coincide or not? The results, I mean.
But you'd hope so. It obviously depends exactly how you measure it. So you always have the
Is anything theoretical known about these different methods, that they should coincide, or?
Well, I guess it depends what you're doing. If you have two different ways, say, to estimate a Bayes factor, they ought to give you the same answer, because it's the same thing you're trying to find. Yeah.
But if you instead interpolate and have a credible interval where you remember your power or the I, would that be the same thing, trying to estimate a credible interval and having a Bayes?
Yeah, I don't know.
one would have to think about that each time, then compare different assessment methods.
The last question, Brian.
So I've probably been done here, but that was a wonderful talk. The bit right at the end about the mixture model and the alpha. So if I think about nonlinear epidemic dynamics of recurrent epidemics, there's probably you'd have to be careful, wouldn't you? Because they might be models that looked very different, but if you let the parameters move around, then they could have identical behavior. So how do you deal with that in that context? So an SIR model can look very much like an SEIR model, for example.
Okay. Well, I guess in that particular method, all that's really going on is that you're literally assessing the kind of likelihoods, and you're sort of comparing one with another. So, that's.
They might be identical.
Yeah, is in some sense. Yeah, it comes down to when I talk about a model in that sense. What I really mean is what is the likelihood of the data I observe given that model, and those are the components that go into it. Okay.
Okay, fine. Well, thanks very much.